Till what step we have done yesterday?
Ma'am, the adapter step.
Okay, means we have trimmed the adapter and the overrepresented sequences using the fast P tool,
Yes, ma'am.
right? Now, what will be the next step? Can anybody please tell me if anybody remembers?
Ma'am, alignment of the reads.
Okay, but here we have to go for one more step, isn't it? Like one more step is there. Just give me a minute. I will
Like,
show
ma'am,
this.
quality check again.
Yes, exactly. Because here, if you will see, we have trimmed the data, isn't it, using the fast P. Now we have to check whether that particular sequences are trimmed or not, whether this sequences got trimmed or not. That also we have to check it, right? So that is what the next step we are having it here, and that is nothing but again we have to check the quality of the raw data or the trimmed data. So this is what we are having the next step. So step 4, is again check the quality of the data. And how we can check it using the FastQC tool. So FastQC, we have seen the command for the FastQC previously only. We'll first of all call the tool FastQC and then we will be providing the output file names. Now here only we just have mentioned the output file names. You can take it directly from here. Otherwise, open the WSL from your respective folder and try to copy paste the names from the WSL. That will also work. It's totally your choice. But make sure you are providing the trimmed file names, okay? I have provided the output file names in the previous step as fastp, that ID. So that's why I will be providing these file names. So let's check it out the data, quality of the data. This WSL I have. Wow. Let's open it from that. Okay, from this folder, I will open the WSL and let's run the step again. It's of 500 MB. Maybe what I can do is I will just compress these files and I will share it with you then if you want to practice it out right now.
Ma'am, also if I open command line, no, like the Linux icon is not visible there.
Okay, what happened to that installation? This particular icon or which one?
Yes, ma'am.
Okay, have you deleted some files of that, like Ubuntu or that files?
No, ma'am.
Okay, maybe you can restart your system then. If issues are coming, restarting is the best option.
Okay, ma'am.
Okay, so here you can see I just have run the step of the fast QC, that is the quality check again for both of the trimmed samples. And now here I'm having the step to be done. I'm having the next part. I can check the file names by ls, and you can see I'm having the HTML and the zip file generated for both of the trimmed files. Now again what we can do is we can check the quality of that uh, files again. So here I'm having the results to be displayed. Fast P underscore 1 and underscore 2. That is the reverse file. Let's open the first file. And from here, I'll just open the previous file also, so that we'll be able to know whether the sequences are trimmed or not. So if you will see before the trimming, here in the overrepresented sequences, we were having two sequences, right? Which are having the possible source as a something primer sequences or adapter sequences, right? Here you can see RNA PCR primer. And here also in the adapter content, you were having this blue colored means what? This is the Illumina small RNA 3' adapter. But now if you will see the trimmed file, after trimming, still overrepresented sequence is showing the cross mark. But here if you will see that possible source, we are not having that sequences available over here. All the sequences are having the no head, means whatever the sequences we have provided for the trimming, that sequences have been trimmed. Right? You can see all the sequences are having the no head. And in the adapter content also, if you will see, that particular Illumina 3' adapter is not present. That graph is removed from here. That's why again green color mark is coming. Right? So this is how you can see your results. Again, if you will check it for why still cross mark? Because ma'am, another no head sequences are there. That's why. Another overrepresented sequences are there, but as no head is there, so we will not consider it. So due to that only you are having that cross mark. Yeah. So if you will check the second file also, the same thing will happen. There also whatever the overrepresented sequences from the reverse file also, we have mentioned the two overrepresented sequences and one adapter content. And if you will see that particular sequences before trimming and after trimming, that will be removed from your data, right? That is what interpreting. Now we are having a good quality data with us. Now we can move to the next step. Okay. So I'll just make it here the zip file for this and I'll share it with you. So that you can also try to perform the next steps then. This zipping is also taking a lot of time. Anybody of you is having the zip file of it? Like we have downloaded this file into our systems. So before extracting it, if anybody is having the zip file for this raw data, can you please share in the chat box so that Pritesh sir will be also able to perform the next pipeline even though the first steps are missed. Because I have extracted the file, so here I'm not having that zip files with me.
Ma'am, it's okay. You can proceed further. I will look into it.
Okay, fine. Not an
Yes.
issue then. So let's go for the next step now. So now here comes our main important step, that is what we are having, the alignment of this particular reads onto the reference genome, right? So step 5 we are having
Excuse me, ma'am.
Yes.
Can you share this step for the fast QC fast P that in the chat box? Because I
Okay.
couldn't copy from. It's not my copy paste is not working.
Okay. Okay. Fine. I'm sharing this command in the chat box. If your copy paste is not working, you can write down the command also in your WSL then.
Okay.
Okay. So now we are performing the next step, and for this step we are going to, for the alignment step, obviously we are going to require the reference genome which we have downloaded in the previous steps only, in the previous sessions only. So now let me just try to write down alignment of reads. Reference genome. But if you remember, like whenever we are performing this alignment, every time we have to run the two steps of it. Like we are having two sub steps for this alignment. First sub step is that we have to first of all perform the indexing of the reference genome, right? Why we have to perform the indexing? Can anybody please tell me? And for this alignment, we are going to use the BWA tool. So that's why for the indexing also, we have to use the same tool, BWA. Okay. So indexing means what? Right now, we are having a small reference genome. It's a chromosome number 17. But in actual terms, whenever you will be running it, your reference genomes will be two-way larger. And as it is going to align each and every base on of the reference genome with the reads, so it will take a lot of time. And if your reference genomes are having a larger size, obviously it will take a lot of time again, right? So that's why if you are performing the indexing means what? Your reference genome will be converted into the short binary files into the binary language, and we know that our systems or the computer, or you can say the tools, can easily understand the binary language easily, right? So just to reduce this alignment time further, we have to convert our reference genome into the short binary files, or the index genome, you can see, right? And if you are using any another tool for the alignment, so you have to perform the indexing with the help of that tool only. Do not use index for the indexing different tool you are using, and for the alignment you are using different tool. It is not going to work properly. Okay, so now we'll be using the BWA tool for the alignment. So that's why for the indexing also we'll be using the BWA only. So now let's try to open the BWA manual. We can open it by BWA. And if I'm just typing the BWA, still my manual is opening. If you are using -help, that will also work. So as of now, further, we want to index the reference genome. So that's why here you can see usage is being provided. You have to call the tool BWA, then different options you can use it over here. So first of all, command is there, and in the command, different options you can use. So here, as of now, we want to index the sequences in a FASTA format, which is present in a FASTA format. So that's why we'll be using the BWA index command. Okay. Rest of the commands I will tell you later on. So let's check the different options in the BWA index now. So BWA index, we can open it. So you can see in the BWA index, we can use these many options over there. So -A, -P, -B, -6 is also there. So -A, we are going to use -A option, which is what BWT construction algorithm. Actually, this BWA is using the two algorithms, which you can see it here, BWT SW and DIV. Now, when you have to use BWT SW and DIV, so you can see here only the description or the warning is provided. If you're using -A BWTSW, it does not work for a shorter genomes, while -A is. And if you're using -A DIV, it do not work for a long genomes. Means whenever you are having a shorter genomes, like maybe if you are having bacteria or if you are having the virus genomes, you can use this -a div algorithm. Okay, but if you are having the eukaryotic genomes with you, you are having a larger genomes or the longer genomes, then simply use this BWT-SW algorithm. Is that clear? So most of the time we usually follow the steps for the eukaryotic genomes. So that's why here also we are having the chromosome number 17 from the human. So that's why we'll be using the -a BWT-SW. Okay, is that clear? And then we'll be using this, like we have to provide our genome file name. So in the raw data, if you will see, I'm having my reference genome here as a chr17.fa. I'll be using the -a BWTSW, and previous to it, I have to use the BWA index options. So let's try to perform it. Let's call the tool BWA. Index is the command. Hyphen, with the help of -a, you are providing the algorithm BWTSW. And then provide the file name chr17.fa. Let's copy the command and let's perform the indexing. I'm sharing this command with you also in the chat box. Okay.
Ma'am, BWTSW is for?
Is for a short, longer genomes.
Oh, okay.
Yeah. For eukaryotic genomes, you can have. So as we are having smaller reference genome, this indexing will happen faster. But if you are having a longer genome, like a human, it may take 4 to 5 hours also, or maybe it can take 1 day also. It will take a lot of time according to your organism genome size. Okay, let it happen. Till then, if anybody is having any doubts, you can ask me and run this step into your system.
Uh, Yesterday we had wrote down some sequences names, right?
Sorry? See,
Sequence 1, 2, 3, 4, like that. Oh, what was that
that
for?
was the FASTA file which we have used for the trimming purpose.
Okay.
The
We have noted it. Why do we note it down
because
in the
that
notepad?
sequences we have to trim it, na? We have to clean that sequences from our data because that was representing the bad quality of our data. So we have removed
Yeah.
that sequences. Here you can see these sequences we have trimmed
Yeah.
down.
Okay.
Yeah, and from where we have took these sequences from the fast QC report.
Uh, okay.
Yeah.
Yeah,
See,
fine.
in this particular, this was my original raw data file. Here I was having that overrepresented sequences, which you can see it here. These
Yeah,
are,
yeah.
yeah,
Yeah.
so this sequences we have trimmed with the help of that file and command. And now we are having this file where that sequences are not present.
Actually, we only use one command, right, to trim
Yes,
down.
yes. For
Yeah.
forward and the reverse file, we have to use only one command.
Yeah. But we wrote down these commands. I mean, these sequences in a separate one.
No, no. In the same file, we have provided the sequences from both of the files.
Okay. I noted down this in another notepad.
Oh, okay, okay. No, in the same file, you can mention the sequences.
Okay, let me see if that is done correctly or not.
Yeah, okay, okay.
Let
Ma'am,
me
is
check
it
if it's correct.
necessary that forward and reverse strand should be like the over represented sequence will be trimmed simultaneously?
Yes, actually you have to mention it in same command if you're using a different tools also, because what happens, if this particular sequence, obviously it will be not present in the reverse strand, right? So it will be not trimmed from your forward strand, right? Because in the same sequence, if it will be not present, it will be not trimmed only. So that is what the thing. In the same file, you can mention all the sequences. And
Okay.
even though if this sequence is present, this is what the adapter sequence is, isn't it? So if this
Yes.
adapter sequence is present in your reverse file, that will also represent the bad quality, right? So even though
Yes.
that sequence is trimmed, your data quality will increase only.
Okay. Okay.
Yeah. Okay. So till then you can see my indexing part is also done. Let me know if you're having any errors in your indexing. If any errors are there, we can sort it out and then we'll move to the next alignment step.
Excuse me, ma'am. I checked mine. The new one. Still there. It is there.
Okay, can you please share your screen?
Yeah. Okay.
Okay.
Mm,
Uh,
This one.
okay, open that file and open the previous file also.
Okay.
Before trimming file also.
This one is after trimming.
Okay, have
The
you mentioned the sequence into your file properly?
No, no. Actually, I
Maybe,
think I
I
missed it.
think, yeah, maybe you have mentioned the reverse adapter sequences into the forward file. Maybe that thing may happen.
Actually, I copied that in a notepad. I didn't know how to relate this one.
Okay. So what you can do right now is just close this all files. You can delete these files also because the sequences are not trimmed from here.
This one.
Yes,
Mm, From where I should delete?
you can delete all the fastp files which are present because your trimming step is not done properly. So that's why quality is also not good. You can delete all those files, fastp files.
This one also.
Yes. Yeah, that one also. Now open your ERR_1 FastQC HTML report. Yeah, that one. Yes, open this one. See in the overrepresented sequences. Open that. And open your reverse file also. Reverse file FastQC report, yeah. And from the overrepresented sequences, now we'll be mentioning whichever the sequence you want to trim in the adapter.fasta file. So in the WSL, create one adapter.fasta file. Go to the WSL right now. Use
Okay.
the touch command, adapter.fasta. Yeah, enter. Okay. Manually go to your folder in a DNSSEC folder and open that adapter.fasta. A second
Uh,
file, second. Yeah.
okay.
Previously, you have generated the text document. Maybe it was not taking that file. Here mention
Okay.
that sequences which you have to trim
Okay.
now.
Yeah.
Okay.
Okay.
And provide this file into your command further. So it will be trimmed.
Um. Okay. Fine.
Yeah. You can do it right now. It's 2 to 3 minutes work. You can do it. Provide the greater than symbol in the note that file. Adapter.fasta file.
Sorry.
In your adapter.fasta file, provide greater than symbol and write sec1, which is sequence 1.
Okay.
Yeah.
Yesterday
Enter.
same will be it will be the same yesterday's, right? So can I
Yes,
copy
yes.
this?
Yes, yes.
Okay, let me see. I have write down somewhere.
Not
Not
this one.
this one.
Maybe in that folder only you are having one file. Go back once to your DNSSEC folder. First file, which is that adapter content from.
Yeah, this one.
Yes. Yeah,
Okay.
save it.
Now?
Yes. Save this. Okay, close it now. And now use yesterday's command in the WSL. Copy paste the
Okay.
command. Yeah.
I think I have that command. This one.
Write down the commands into the pipeline.sh directly, okay? Oh, okay. In this FASTA files, do not write the commands, okay? In the FASTA file, only your sequence should be present, okay?
Okay.
As this was a text document, that's why it was not taken by the WSL. So not safe.
Okay.
Yeah, enter it.
This one.
Okay, let it run.
Okay.
Your file names are correct, right?
Yes, yes.
Okay, fine.
This one you shared yesterday. This.
Yeah. Okay, so let it run, and after the process will be done, again check the quality and check whether that sequences are trimmed or not. Okay, you can
Okay.
stop your screen share now.
Okay.
Yeah, do not close that terminal. You can go for the next steps also. Okay, if you want to run the indexing steps, you can open the another terminal and you can follow the steps further.
Okay, I have to run again this BW index in that step.
No. You have run it previously only, right?
Yes.
Yeah, no need to perform the indexing again then.
Okay.
Yeah. So I'm sharing my screen again and let's try to perform the alignment now. So now again for the alignment, we'll be using the BWA tool. That is what the second step we are having it here. Yeah. So the second step we are having the alignment of aligning the reads on indexed genome now. And here, if you will see, I haven't shown you the files. If you will see here, previously we were just having the chr17.fa, but after running the indexing step, we are having these last 5 files generated. The number of files, indexed files to be generated will be totally depends on your system and the organisms, genome size also. Okay, maybe 8 files will be generated, 7 files will be generated. It depends. So yes, here we are having the 5 files. Let's go for the next step now. So what will happen actually? So now we are going to align the reads from our this file onto these many indexed files, right? One by one. So for that, we'll be using the BWA tool again. So we will call the BWA tool, and in the BWA, we will be using the MIM algorithm now. Let me again open the BWA. And from here, we are going to use this BWMM algorithm for the alignment further, because for the alignment, for the indexing, you were having directly command, but for the alignment, you are not having direct that particular thing, right? So here you can see, actually, there are 3 alignment algorithms in the BWA, MIM, BWASW, and this is the one. But out of all them, we'll be using the MIM algorithm right now. Okay, description for each and every algorithm. Again, what you can do is you can open the manual for each and every algorithm and you can just check it out. Here also the thing is provided. If you're having BWA, SW means what? It's for a long queries. If you're having a shorter queries, no need to use it. If you're having this SMSE, SMPE, so it will generate the single ended or the paired ended data. Okay. As of now, we'll be using this MEM algorithm. So let's try to open the manual for the same. BWA MEM. And in this particular BWMEM, again, a lot of options are provided. So now which options we are going to use it? So we'll use these things. Then options, we are going to use the threads because if you are providing this number of threads, it will reduce the alignment time further. If you are having a longer genomes, it's better to apply. Always you have to give the threads to the process. So what will happen if you're having the 4 processors into your system, you can provide the 3, maybe 2 or 3 threads. Do not provide all the threads for one step only. Otherwise, your system may slow down. So provide one thread less than your actual processors. Okay. So we will be providing the 4. Most of the time, you will be having the 4 processors. So we can provide 2 threads over here. Okay. So that's why we'll be using the -T option. Then we are going to provide our index file name, which is what the chr17.fa. We are not directly providing the indexed genome file names. We are not going to provide these file names because it will be required at the backend steps. Okay. Even though I will provide chr17.fa, but at the backend, these files will be taken by the tool and it will perform the alignment further. And then we'll be providing the input files, which you can see it here. Here you have to provide your index file name, your genome file name, and then here you have to provide your input FASTQ files. And then maybe if you want to provide the output file name, that you can mention it with the help of greater than symbol. So let's generate the command now, bwa mem. Then we'll be providing the threads as at the number of 2. Then this is what I'm having the genome. Then let's provide the input file name. So we are having this input files with us. Make sure you are providing the trimmed files for the alignment further, okay? From where your data is trimmed properly. And then we want to generate the alignment file separately. So you can provide the greater than symbol and then you can provide your file name. So alignment file name, output file name, you can just provide it according to your choice. Just for the understanding purpose, I'm just providing the file name as a BWA so that I can understand this is the alignment file. So I'll be providing the file name as BWA. And can anybody please tell me whether 2 output files will be generated or only 1 file will be generated? How many outputs should be generated here now?
Ma'am, only one.
Yes, exactly. Only 1. Because what happens in the forward and reverse file, you are having the same data which is sequenced from both of the directions, right? So even though you will align your data from the forward or reverse, it will align at the same location, isn't it? So that's why we will be generating only 1 file over here. So let's provide only 1 file name. If you want to generate the SAM file, you can provide .sam extension. If you want to generate the BAM file, you can provide the .bam extension. Okay, SAM and BAM, both of them are alignment file, standard alignment file formats. So I can copy this command from here. I'm sharing it with you in the chat box. And I will just run it in my system also. And let this alignment happen. It may take a bit time. So let's wait for it.
Excuse me, ma'am. For me, this HTML didn't come.
Okay, you can share your screen then till the step is proceeding. HTML file will come when you run fastqc. For that file, when you check the quality for that file, then only your HTML file will come, right? So type fastqc
Okay.
space, just type fastp underscore err. Press tab, it will take that ID automatically. Underscore one, type one. Underscore, I think you have provided 2 times.
Just
Backspace once. Underscore one. Provide tab again. It will auto detect your file. Enter it. Now your output files will be generated in terms of HTML and the zip. Later on, run it for the reverse file also.
okay.
Yeah. Yeah, here you can see your files are generated.
Yes.
Okay, run it for the second file also. You can use up arrow key also to use the previous commands.
Okay.
Instead of underscore one, provide underscore two. No, your arrow, that cursor will not work. You have to use, yeah. And now check if the sequences are trimmed or not. It will be trimmed.
Okay.
Open that file. Third last file. Yeah. Go to the overrepresented sequence. See, these
Yeah.
are.
Yes. Yes.
Okay.
Okay.
Okay. If anybody is having
Yes.
any doubts in the this fast QC or maybe in the alignment, you can also share your screen, or maybe if any errors are coming, you can show it to me so that we can resolve it.
I'll stop sharing.
Yes, you can stop it.
And now I have to do this from again, right? BWI index.
Indexing is done, so no need to do it again. You can go for alignment process directly.
No,
And if indexing is not done, then go for the indexing.
I think I'll go for this.
Okay, fine.
And
Yes, Janvi ma'am, Piali ma'am, are there any doubts from your side? So alignment process is being processed. It's proceeding, so we have to wait for some time. Let me know whenever your alignment will be done, okay? Okay, if any doubts, errors are there, please let me know. I'm waiting for your queries only. Otherwise, the next step we have to do it tomorrow only because this indexing will, this alignment will take a lot of time. So if any doubts are there, if any queries, okay. Here you can see my file is generated, my alignment is being done. So now I'll show you just the another command by which we can convert this BAM file into the SAM file. Because if you will see here, am I audible?
Yes, ma'am.
Okay. So now here, if you will see, this BAM file is generated, right? It is having some size. But what will happen now, if you want to see how actually the alignment information is getting stored into this file, as it is present in a BAM binary format, you will be not able to understand it, right? So you can convert this BAM file into the SAM file format with the help of SAM tools now, right? So we will just convert it and then we will stop it, okay? I'll just show you the command directly. So the next step we are having as conversion of this BAM file into the SAM file. Conversion of BAM into SAM using SAM tools. So again, first of all, we have to call the tool SAM tools. Again, you can just check it out the manual, okay? All the options along with the description that will be provided in that. Maybe if you are not having it in manual, you can just check it, the manual online also, how to use the SAM tools, okay? So as we just want to view this BAM file into the SAM file, so you can use SAM tools view option. Just provide your file name, that is BWA. This is your input file name. Provide that BAM file name and just provide this greater than symbol and provide your SAM file name. So I will provide the same file name, but here just the extension should be SAM. That's it. My command is ready now. I will share this command in the chat box. Copy paste it somewhere so that it will be there with you. So after the alignment, you can also try to run it. So Control C. Okay, it's running. Now this conversion will take some time. We have to wait for that. Okay. So tomorrow, I hope you will generate this SAM file and then we will see the next step. Okay. I will show you the alignment information, what actually is stored in this alignment files. Is that clear? If any doubts are there, you can ask me now.
I gave this one, but nothing came. The next. Can
Which
you share my screen?
Yes, you can share it.
Yeah, this one.
Your alignment process is done or not?
Yes, it's done.
Okay, means your SAM tools view. See, SAM file will be generated into your system then.
Okay,
Okay.
this one is. Oh, yeah, okay. I thought there's something else.
Okay, where is your SAM file? Yeah, third file is
This one. Okay.
process generated. Okay. Do not try to
Okay.
open these files manually because these are having a lot of data inside it. So I will, with the help of commands, you can revise the Linux command. You may use the head command to see the content. Okay. Tomorrow
Okay.
I will tell you in each and every column what information is present.
Okay.
Okay. So yes, then that's it for today. Let's meet tomorrow for the next steps then. Bye bye. Good night.